Okay, we left off on Monday talking about skeletal muscle structures and how those structures impart function. Okay, we left off talking about when an actin and myosin molecule bind together, it forms something called what? A cross bridge. Okay, all force production I shouldn't say that. Most force production results from the formation of cross bridges. Okay? If you are stronger than someone else, which we'll talk about on Monday, you are likely able to form more cross bridges than them. Okay? That's where this is going to come from. So, once we get a cross bridge that forms, I told you guys today, myosin is a molecular motor. Myosin grabs the actin and it wants to try to pull in one very specific direction. Okay? If it is able to do that, that executes something called a power stroke. Okay? The power stroke. Power is, anybody had physics? What is power?
Work over distance.
Force over distance. We're close. I think that's actually work.
Oh, work over time.
Work. So it's basically force over time. Those are all kind of related. So it's force, time, or force and velocity. Sorry, which is the thing. It's distance and time together is velocity. Okay. So that's where power comes from. So we are applying force from the head. We're moving and it moves at a certain speed. That's where we're going to get the idea of power. Okay. All concentric muscle contractions. Bicep comes up. Okay. All concentric contractions are the result of myosin heads undergoing a power stroke. Okay. We're going to keep that in mind. This is really, really important. As this happens, actin molecules, those thin filaments will slide past each other. Either side of the sarcomere, they're going to pull inwards, in on both sides. Okay. And this is great. But now we have a problem. Okay. That only moves us like tiny, tiny, tiny fractions of a centimeter. Okay. Myosin's not very big. So I grabbed on and I moved, and you guys could undergo a power stroke, like one single solitary power stroke from all of the myosin in a muscle, and you would not even perceive that you would move. Okay? Because the distance traveled is so short. Okay? So what we have to do then is disengage the myosin, move it so it goes here and it pulls. We have to get it to let go, move back like this, back to its original shape, grab on again, and then pull again, and let go, and go back, and grab on again, and pull over and over and over and over again, thousands of times, in order to generate what we perceive or what we can actually see as movement. Okay? So we're going to call this process, this bind on, power stroke, let go, go back to the start, grab on again. Okay? This is called the cross bridge cycle. Okay? The cross bridge cycle. Of all of the things that we're going to get with skeletal muscle that relate to movement, this is one of the absolutely most important things that describes or underlies movement, force production, and energy use during a particular movement. Okay? It all comes back to this very simple four-step process. Okay? Bind, power stroke, let go, go back to the start, or sort of re-engage. Okay? And do it over and over and over and over again. All right? Because of that, I'm always loath to ask you guys to memorize things But in this instance, you're just going to have to memorize all of the steps. Okay. Here we are at rest, right? No contraction. Troponin and tropomyosin are where they're supposed to be. Myosin heads cannot bind to actin. The muscle is relaxed. We get calcium release from the SR. It binds to troponin. Tropomyosin moves. Boom, myosin can engage, right? It engages, and then it is going to pull, and it's pulling from both directions here, and we slide. Now that it's here, now we have to get it to let go. Okay, we've got to get it to let go. What I am about to show you guys, to me, and there's a lot of weird things in physiology. What I'm going to show you, what I'm going to explain to you, how this works is arguably for me the most counterintuitive thing in all of physiology, or at least in most of exercise physiology. Okay, so it's very simple. What you guys need to memorize and what you need to understand is what is happening between the actin and the myosin. Okay, and then on the actin molecule, we need to understand, do we have ATP, ADP, and inorganic phosphate bound to the myosin or not? Okay. The state of binding of ADP and inorganic phosphate and ATP determines what we're going to call the energy conformation of the myosin head. And it's the presence or absence of those things that will denote, are we basically, it's in one conformation right after it's done a power stroke, and then in another one when it's ready to be able to bind back up again. Okay. So what happens in the cross-bridge cycle? This is where a majority of the energy that you expend doing any movements, any exercise, any physical activity comes from this cellular process. Okay. When you walk, you do this process a certain amount, it costs you a certain amount. When you run, you do this process many more times. So it costs you more energy. Okay. That part is simple. It's the point where you use the energy that's just fucking backwards. Sorry, it's just backwards. Okay. Let me walk you guys through this, and I hope that it will then make sense. What you guys see right here in step one, okay, we're going to call step one, the myosin head is in what we call its high energy conformation. Okay. We have just opened the binding site on actin. Myosin has bound and it's grabbed onto the actin and has not yet done a power stroke. On that myosin head is a molecule of ADP and a molecule of inorganic phosphate. Okay. We'll get to in a minute where that has come from and why it is there. But just let's start with the fact that that's what exists. Okay. It is here. It's attached. Okay. The next logical step makes perfect sense. The myosin head tilts and it pulls the actin molecule. In doing all of this, it lets go of the ADP and the inorganic phosphate. It releases them. Okay. Now it is in this, it's done the power stroke, it's released all of this. We call this its low energy conformation. Okay. It's low energy conformation. Here's where it's weird. Okay. The fact that it just released the ADP and the inorganic phosphate. Okay. We did a power stroke and we didn't actually use any energy. Okay. We had used the energy before we ever got there. And we just let go of those molecules. Okay. So here's the backwards thing. We got low energy conformation. It's bound. Here comes ATP. Here comes the energy we're going to use. It binds to the myosin. And the myosin then detaches from actin. We're still in our low energy state. We still haven't technically used any energy. It detaches, and then the myosin ATPase enzyme that I showed you guys that exists right in the head of the myosin molecule when I was showing you the light and the heavy chains on Monday. That myosin ATPase enzyme now hydrolyzes or breaks ATP into ADP and inorganic phosphate, which then stay attached, and the myosin head goes back to its high energy conformation. And now it is detached, it has these two things bound, and it has moved back into sort of its topped position, and it's ready to go again. So the backwards thing is we don't use energy to generate force. We don't use energy to generate the initial movement. We use energy to get myosin to let go of the actin, and then to move the myosin head back to its starting position so it can generate force again. Okay? So it's a very, very weird, to me, it's very backwards. You don't actually need energy or use the energy during the power stroke. You use it to let go. Okay? And so that's why, and that's going to inform a whole bunch of other things that we're going to see and talk about next week. The fact that this doesn't cost me energy to get it bound and form the cross bridge is why running or walking downhill or going downstairs feels so much easier than going up, because it costs us less energy. Because we don't have to run the cross bridge cycle many times and in the same way as we do when you do concentric contractions. Okay? I promise you, this in this exact form, steps, ADP, inorganic phosphate, the myosin head, its energy conformation, all of this will be on the test. Okay? We have to understand this. Okay? We have to understand this in order to understand once we start moving. Okay? Once we start trying to put force into use, why things look certain ways. Okay? All right. Questions? Comments? Concerns? No? We need to make some people do some eccentric contractions while those are doing concentric contractions, and we'll see who fatigues first. No? Yes?
You mentioned earlier about how, like, when one myosin kind of does the contraction and moves it, it's not a noticeable difference. So is what causes a noticeable difference, like the myosin head moves the actin, then goes to another actin, it just keeps like a tug of war?
Yes, it is exactly right. This is a tug of war, okay? And we will talk about that even more next week. But it is pull, and it moves this tiny, tiny fraction. And then you have to let go with one hand and reach back out and pull and let go and reach. And eventually, like, we don't ever think about this in this finite one cross bridge cycle kind of thing. You just think, pick up my phone, move my finger, right? Talk, whatever it is. But there are hundreds and thousands of these things happening while you move through this full range of motion and those things. It happens so fast and in such large quantities that it lets us do all of this, okay? But at its most basic, every myosin head is doing this, okay? And so if you put that together on a very, very large scale over and over again, that's how we get movement. That's how we get these. Okay. Great question. Anybody else? Yes.
Is calcium being released when I'm ATP?
I'm sorry, is calcium being released? No, you're not releasing it. So,
When does it release?
okay, great question. So the question is, I've done this once, we've hydrolyzed, I've broken everything apart. In order to get movement, I, in essence, need this to start over again right now, okay? I've done one cycle, I didn't really move very far, so I need it to be able to reattach immediately. Great question. So are we releasing calcium again? And the answer in many ways is it's not really being released again. It just hasn't ever gone anywhere. You've released the calcium and it still exists out in the cytosol basically, so that there is still, this happens so fast that there's still going to be calcium bound to the troponin, so the tropomyosin is still going to be pulled out. So in essence, as soon as this goes back to high energy, it will bind again almost immediately. And it can go over and over again. And now eventually in time, if you don't keep getting action potentials, you don't keep releasing calcium, you pull the calcium back into the SR, it will eventually then not be on troponin and we will begin to cover up the binding sites and we can't run the cycle again. Does that make sense? I don't know that I actually explained that in the, we will come back to this, okay, a little bit more so later today and some on Monday, and we'll try to put all of the processes before and after this sort of together in a time sequence, and we can talk about kind of what that looks like. Okay, but that's a really, really good question. Okay. Yes, ma'am.
Is the inorganic phosphate and ADP released at the same time?
No. I mean, it is important, but for what I want you guys to know, it's not important. It's important to know that they both get released. They are technically, one gets released and then the thing happens and then the other one gets released. And inorganic phosphate is a weird thing that causes us all sorts of problems at various points. So they do not get released at the same time. It's not simultaneous and different things drive each one, but it's beyond the scope of what you guys need to know. Okay? Other questions? These are good. These are good. Okay. All right. Now, let's talk about fiber types. You guys heard of muscle fiber types? Y'all cover fiber types in anatomy or in physiology? One of those classes? Or you probably talked about it. You guys have all taken all your elective classes, right? Beforehand. We're going to fix that coming soon to an HES department near you. This will be the first class that people take and everything. We're going to fix this. We're going to be a school next year. Did you guys know that? We're not going to be a department anymore. We're going to be a school. And there's a program out of the dean's office called Population and Community Health or Public and Community Health, and they're going to be in the school with us. And then in another year after next year, basically, we're getting rid of the enrollment when you have to apply. Like, if you just want to be an HES major, you get to be an HES major. This will be, we're going to redo our sequence. This will be first. You've passed biology, you've passed chemistry, you've passed anatomy and phys, you have your prereqs, you take this first. Then this class unlocks the other ones. We're going to have tracks and various things where you can take more ex-phys classes or you can take more health promotion classes, depending upon what you want. And everyone's like, I know. So changes, they are coming, kind of down the line. So my guess is you guys have seen this in the personal training or endurance training or one of your other ones if you didn't have it in anatomy or something like that, right? Okay, somebody, what is a fiber type? Anybody knows?
There's 3 fiber types and they determine many different things about the muscle action and
Okay.
its capabilities.
Okay, there are 3. Can you name them?
X, type 2A.
Type 2X, type 2A, and type 1. So what the hell are those?
Type 1 slow twitch and then
What's that?
means that there's like small in size and they typically don't like produce as much force, but they're the most aerobic capacity out of the 3
So why are they called slow?
because they take like they don't fire as fast as type 2 and A and X.
Okay, there's a lot of truth in all of those. We're getting mixed around a little bit, but that's okay. All right, that's okay. Muscle comes in different flavors. That's what I like to think about. Okay? I'm sorry. You guys like to eat hamburger and steak and chicken, you know what you're eating? Muscle. Okay? I'm sorry, it's muscle. I want to be a drag on a Wednesday. If I could be, I'd be a vegetarian, but I do not like vegetables. Right? I like Doritos and not vegetables. Okay? My daughter is basically like Greek. She would eat like a charcuterie board all the time. Like her favorite things to eat are olives, right? Oh yeah, she loves olives. She would eat olives and like fancy cheese and salami and, you know, blueberries, like that would be her favorite thing. Anyway, I'm sorry. Muscle has flavor. There's a reason, right? You guys at Thanksgiving, for Christmas, you have turkey. There's white meat, there's dark meat. Guess why? This, okay? Literally different flavors. Okay? All right. So it's technically not that the muscle comes in a different type or flavor. It's the myosin that comes in a different type or a different flavor. Okay? Not all myosin are created equal. Okay? So we began to understand all of this in humans at least. This is stuff that we had known in animal models of things for years. Okay? But in humans, through the advent of the muscle biopsy, okay? We make an incision, we stick this Bergstrom biopsy needle in, you apply suction on one end, you pull some muscle fibers into this little window, you slide the plunger down, it works like a guillotine, and it cuts off, it doesn't actually sit perpendicular like this, it's actually in the needle. But it sits like so, and then some poor grad student has to tease it out with a pair of pliers, and you affix it onto a popsicle stick, and then you dip it in liquid nitrogen and you freeze it, and voila, we have muscle. Okay? Does it hurt? Well, it all depends, right? So generally when these are done, at least when we did them, you get a local anesthetic. And so anyone that's ever had local anesthetic, right? They've done an injection in your mouth because you had to have filling or a crown or something, or you've had anything else done. I mean, sometimes getting the local anesthetic is not always the most painful thing, right? Like there's a needle, they shoot xylocaine or something in, it kind of burns a little bit. What it feels like once that part is done is it just feels like pressure. Okay? Now, it is unpleasant in a couple of hours when all of that wears off. Yes. Right? I mean, you have a literal hole in your leg. So it's pretty sore for a few days afterwards. Also, you got to like wrap it up in saran wrap, and when you get in the shower and things, there are a lot of procedures with all of that. So that is on a calf. Typically, we would take them on the vastus lateralis because there are no major blood vessels or nerves that you can hit out here, but you can take them from any muscle. Okay, so you get this little baby piece, and then you stand it up on its end on the popsicle stick like this, where you can see the individual fibers, and then you cut these tiny, tiny little cross sections. So when you look at it under a microscope, you see each individual fiber, you see each individual little cell in the way that it's going to look. Okay. And so it looks, let me get down here, it kind of looks like this. Okay. You're looking into it and you see the cells that look this particular way. Okay. Now, one of the ways that we know that there are differences in these things has evolved over time. Okay. And so we originally thought there were two kinds of muscle. There were two flavors, fast twitch and slow twitch. Okay. So when we say fast and slow, what we mean actually is we are describing how quickly it goes from resting to turn the fiber on and force production. It does a power stroke, force production goes up, then we shut it off and it comes back down. So how steep basically is that slope of the rise when force gets turned on? Fast twitch fibers, that rise is fast. I mean, they're both really fast, but it's faster than in slow twitch. Okay. Fast versus slow is really a convention that came from animal models of doing these things. In a lot of ways, before we had really good microscopes. Okay, you could basically cut out a muscle, string it between two things, zap it with electricity, make it generate force, and you can figure out that some muscles turned on and went up faster than others. Okay, that's where fast and slow came from. So as we began to get better with microscopes and chemistry, we did something or learned to do something called histochemistry. Okay, histology. You guys ever heard of histology? No. Some of you, yes. A few, yes.
Maybe organs sometimes.
Sometimes tissue and organs, right? So we can use histology to try to figure out, like, what do these cells look like? What, do they have this membrane and that membrane? So we began to take these individual fibers, okay? And if you incubate them with ATP and you put them at different pHs, or you stain them or you incubate them with something that will bind to glycogen or something in there, we began to see that there are differences, okay? That not even in the fast, especially in the fast fibers, they're not all the same. And so the big thing that we figured out, and this is where kind of the original convention of fast versus slow and kind of the 1 versus the 2s and the 2As came from, is all going to be about using immuno or using histochemistry to look at the myosin ATPase. So it's that enzyme that breaks down the ATP that lets us let go and break the cross bridge and go back to our high energy conformation. It's different, okay? It is different between the different fibers. Okay? So again, now you see why the cross bridge cycle is so important, right? The speed at which we can let go and go back to the high energy conformation plays some role in how quickly we can actually get force to come up or not. Okay, we can also stain for different metabolic enzymes. SDH stands for succinate dehydrogenase. Y'all don't have to know that yet. It's an enzyme in the Krebs cycle. Okay, it's an enzyme in the Krebs cycle. LDH is lactate dehydrogenase. It's an enzyme at the end of glycolysis that turns pyruvate into lactic acid. Hexokinase is the very first enzyme in glycolysis. It puts a phosphate onto glucose and traps it inside of your muscle cell. They began looking at these enzymes, who began looking at the presence or absence of glycogen and lipids inside of our cells. And what we figured out was there are some kind of patterns and differences that exist with all of these things. And I'll talk about what those are and show you some pictures in a minute. Then, remember I told you guys on the first day of class, or the second, we got the first advent of immunohistochemistry. So the technology that we use in tests and COVID tests and those kinds of things that you can take at home right now was first used, when it was a big deal, was used with all of this. So we have antibodies made by the immune system that bind very, very tightly and specifically to a piece of the myosin. Right? It's not just how does the myosin use energy, how quickly does it. We can determine, is there little pieces of all of this that are going to be different? And the answer is yes. Okay. So with the advent of immunohistochemistry, we now look at things that are called the MHC or the myosin heavy chain. So those larger molecules that run along the outside and form the scaffolding of the myosin head. Okay. And we saw that those were different. Okay. We are going to talk about this with 3 primary fiber types. Okay. But that's an oversimplification. There's actually like 10. Okay. I don't know if there's more like 8. But there are these intermediate fibers or hybrid fibers that exist in between because some of them can transition. And then there's some fiber types that we see in animals, and we only see them in humans if you're on bed rest for months, or if you have a spinal cord injury and some things, okay? And that's going to change a bit of kind of what the naming convention has been over time. I will use all of these interchangeably. I'm sorry, okay? but I want you to be aware of what all of them are. So the original things with the observation and the histochemistry was fast and slow. It's all based upon myosin ATPase speed, okay? Fast twitch fibers, slow twitch fibers. As we got better at things, we figured out that slow twitch fibers are all basically the same, okay? They're all basically the same. But fast twitch fibers are different. There are different types or different flavors of fast twitch fibers, okay? So based upon the heavy chain, we call an isoform. So the differences in the heavy chain are going to denote different isoforms, okay? Remember, MHC stands for heavy chain. Then slow twitch fibers began to be called type 1, okay? They're all basically the same. All flows are type 1s. But within fast twitch fibers, which we call type 2, there are what we call 2A and 2X. These three, 1s, 2As, and 2Xs, are the three that we typically talk about all the time, okay? However, there are other ones. There are things that are called 2Bs, which are super fast. Humans have almost none of these, ever. Very prominent in sort of lower animals and things that sprint a lot, okay? Rats, mice, cats, okay? Things like that. They have 2Bs. And then there are what we call hybrid fibers. There are these fibers that sit in between all of these that you have some amount of. We'll talk a little bit about hybrid fibers when we talk about training. Because if you train or you stop training, then a lot of your hybrid fibers actually go away because you transition them and you push them towards being A's or X. Okay? So slow twitch are type 1, fast twitch are generally 2A and 2X. So let's talk about the differences between 2A and 2X. Okay? 2X are the fastest of the fiber. They have the fastest ATPase speed. Okay? The fastest ATPase speed. 2As are a little bit slower, but they're still way faster than type 1. The big primary difference between A's and X's is that A's are much more metabolically efficient. Okay? Much more metabolically efficient. They tend to have much higher levels of mitochondria. And they tend to have more capillaries that go to them. They're much better at doing aerobic metabolism. Okay? And the take home from all of this, the functional consequence of this is, if you have a bunch of 2X fibers, you do like 2 contractions and you're tired. Okay? You are fatigued, right? They got like 2 contractions in them and they're done. 2As, on the other hand, are going to be much more metabolically efficient. They're going to be much more fatigue resistant. And so they can continue to contract for a longer period of time. Now, if you compare sort of the aerobic metabolic profile and the efficiency profile of 2As to 1s, again, there's still a giant difference, okay? 1s are like electric cars or hybrid cars. Very sort of fuel efficient. 2As are kind of somewhere in the middle, right? And then X's are like, I don't know, like a Hummer or like a giant, right, 4-door dually that gets like, 4 miles to the gallon in some ways. Okay. So we'll talk about how all of this plays a role in exercise performance in a second. Okay. A couple of other things to remember. You are born with a particular percentage of your fibers being 1s and 2s. Okay? You were born that way. Thank your parents, curse your parents. The vast majority of us are 60-40 one way or the other on these things. Most people are relatively equal 1 to 2. Okay? You cannot change that. No matter what you do, no matter how much you train, Your 1s and your 2 percentage is determined at birth. Okay? There is an ability to move your 2A's and 2X's between each other. Okay? If you're very, very active, or even if you're just mildly active, your 2X's, all of your 2's will mostly be 2A's. If you're very sedentary, you're gonna have a much larger percentage of 2X's. You're still gonna generally have more of these. X's, the body does not like, they're not evolutionarily helpful to have X's, right? Especially for us. We don't do a lot, like in comparison to other animals, we're very slow. Like we don't have a great need to be able to sprint, okay? Our superpower is that we have great endurance. And we're really, really efficient because we don't have hair, and our weight to body surface ratio lets us dissipate heat really well. They think that back when we were hunter-gatherers, we would chase deer, like just jog after deer all day. And eventually the deer overheats, and we don't. And then when the deer passes out, it's overheated, food. There we go. Okay. We are, in comparison to things, we're very, very metabolically efficient, even if we have some 2A's. I will also mention, there are some slight differences among the muscles in your body in their ratio of type 1 to type 2 fibers. Okay. And that mostly falls along the lines of what is the function of that muscle. Okay. But generally, if you're 50-50 in, say, your quad, you're going to be close to 50-50 in pretty much every other muscle in your body. It's not like there's these wild differences. Your diaphragm is the slowest muscle in your body. The next slowest tends to be your soleus, okay? And then like your erector spinae, postural muscles, and then the diaphragm tend to have the largest percentage of type 1 fibers. You use them all the time, right? Can you imagine how big and strong your bicep would be if you'd been doing curls as many times as you've been using your diaphragm throughout your life? How tired it would be? Diaphragm never gets tired. It's very, very tight. Okay. And then muscles like your quads and your glutes, things that are meant to be kind of explosive that we use to kind of do these very faster contractions, tend to have the largest percentage of 2's, but they're still going to be pretty close. Okay. If you want to be a great sprinter, you got to have a lot of type 2 fibers. I can't help you. Okay. You either have them or you don't. And if you have a lot of type 2 fibers, in addition to be able to run fast, you're going to be able to jump high. You're going to be able to generate, lift more, do better things when you do like power clean, things that require high force and very quick generation of that force. You just have the right myosin and PAs or you don't. Okay. Olympic level sprinters, you're probably 80% or higher type 2. But there's just not a lot that we can do about all of that. Okay. You have to have that just to be in the ballgame. And then once we're there, then running form matters, leg length matters, some things like that are gonna help us a little bit. But I can't really do a lot. You're either a good sprinter or you're not. I can train you to be a little bit better one way or the other. But like I'm never gonna run, you know, I'm never gonna run under like 12 seconds in the 100 meters, much less like under 10 like the really, really fast guys do. It's just a thing. To be a good endurance athlete, though, it's much different. All depends upon your endurance difference, but we see folks that are really good at 3,000, 5,000, 10,000 meters, okay, in those ways. Half marathons. You don't have to be 90% type 1 to be good at those. In some ways, it's actually beneficial at some of those somewhat shorter distances to have some more type 2, because you need some speed in the midst of all of them. Okay, because of our ability to turn the 2A's, to have lots of mitochondria, to be really, really metabolically efficient, we can do some things there. Okay. All right, let's see what else we got here. This is just kind of showing what those things look like. I talked about all of this. The last of the thing to get in it, and I should probably move this until after we talk about the nervous system. But there are some other differences in sort of nervous system side of things that connect to the fiber types. Okay. we will see, we may get to it today, we probably won't because of the quiz. The motor neuron, the cells in your spinal cord that are the relay point between the motor cortex, your brain, and skeletal muscle are all they're all going to have different sizes. And the ones that are going to connect to type 2 fibers tend to be larger, and they're going to have more axons on them, and they're going to have larger, what we call motor units. There's going to be more fibers connected to a single motor neuron in type 2s. This is very key because it is going to play a role in when we generate force, when you do things, it's gonna help us understand why we use type 1 fibers during low force contractions and early on in contractions, and we use the type 2s later on or only during high force contractions. We'll get into that a little bit later. Type 2s have larger motor axons. They're fatter, okay? The axon has a larger diameter, has more myelin in it, okay? Because of that, it has a faster conduction velocity, okay? And so you can transmit an action potential from the brain to a type 2 fiber faster than you can to a type 1, which makes some sense. I gotta get out of the way right now, okay? Those things. So we talked about these things. We'll bring more of this out as we move along and get into those things during class, okay? Here is when we stain the fibers. This is the ACPA stain. So you're going to see the light colored ones are slow twitch or type 1 fibers, right? The black, the very, very dark ones here, there, probably a couple of these are 2As, and then these kind of gray ones here are going to be the 2X fibers, okay? You do this, you stain them, it takes a day, you hope you didn't fuck it up, and then you put them under the microscope, and the poor grad student has to look, and you got to go in and you got to circle them, and you got to figure out here's the border, and then you figure out what the optical density, so how black is it, how dark is it versus these two, and there are some cutoff points. You're like, okay, that one's dark enough, that's a 2A. Okay, this one is light enough, but it's not too light, that's a 2X, and those things. Okay. And so then what you can do, and these are not matched up, but you take these little sections and they're so thin that you get a picture and it looks like this. And then you have the next little slice and it is absolutely identical, but you stain this one for ATPase, and then you stain the next one. This is for a mitochondrial enzyme. So then you can see these, it would look different. You're like, okay, I've got a type 1 fiber. And then in the SDH stain, it is darker or lighter compared to the other ones. And so then that's how we began to learn these fibers have these other sort of characteristics. They have this metabolic profile, they have this many mitochondria, they have this much glycogen, et cetera, et cetera. Okay. We don't do this very much anymore. Fiber typing is not really a big thing that we really need. This is a very kind of 1970s into the 80s kind of exercise physiology type of thing that people have done. And we just don't really use that kind of stuff. Okay. All right. So this is pretty much kind of where we are. So I said that this was genetic. I'll do one last little thing here, and then we'll talk about the nervous system side when we're taking a quiz. We have done a tremendous amount of research in animals. We still do. It's absolutely necessary. But it's also, to me, in some ways, it's incredibly cruel. It's why I cannot ever be an animal researcher, right? It's not that hard to go from that mouse or rat that you're doing things on in the lab to come home and see your cat or your dog and various things in that way. But they've done weird experiments on these things, and then we see it play out in humans in times as well. So if we take a mouse or you take a rat, okay, and you cut, let's just say, the motor axons that are going to fiber types. Cut it open, cut the motor axons, right? Typically what they will do is they'll cut the larger nerve. So lower animals have much more distinct, if you look at their version of the gastroc and the soleus, ours are pretty close in fiber type. In like mice and rats, soleus is super slow, the gastroc is much faster. and they will go in and they cut the axon that goes to the soleus and to their gastroc, okay? And they swap them. And they reconnect the old soleus axon to the gastroc, and the gastroc onto the soleus. And then they wake the little mouse up, and they let it go on for a while. We're not gonna kill it yet. And then they wait, and they give it a few months to recover, and then they sacrifice it. And what they find is the soleus is now a fast twitch muscle, and the gastroc is now a slow twitch muscle. Okay? So what we've learned from all of this is that part of, like, all muscle fibers have the genes that will code to be type 1 or 2A or 2X or whatever they are. But there's something magic about the nervous system, the axon that comes from certain motor neurons in your spinal cord that connect to it, that essentially guide and tell which genes to turn on to make the myosin heavy chain. Okay? We see this in people when you get older, right? Grandma's a little slower. Grandma's not really a great sprinter anymore, right? Grandma's reaction time has gone down. Okay? Part of the things that happen as we age is we accumulate neurotoxin. And neurons in our spinal cord, motor neurons will die. When the motor neuron dies, then the skeletal muscle fibers will also die. Unless they get reconnected from axons of things that are around them. For whatever reason, neurotoxin seems to preferentially affect the motor neurons that go to type 2 fibers. And as you age and those die, the axons will branch off from your existing type 1s and reconnect to them. And you see older people tend to have much higher percentages of type 1 fibers than they did when they were younger. Okay? It's because of this switching that is driven by all of this. And that's partially why grandma's slow. You can't move around. She can't sprint. Her reaction time has gone down. Is that she doesn't have the fast myosin ATPase anymore. Okay? There's other reasons too that go into all of that, but that's one of the big reasons. Okay? Okay. There we go. Any last questions about fiber types? We'll bring all of this back up again as we start talking about function and why some people run faster, why some people have more muscular power than others, like with fiber type, and then some things when we get into metabolism and the cardiovascular system that are going to be different among the fiber types. Okay. Yes, sir.
If younger people have concussions and have like brain damage, do you release neurotoxins and then your body creates more fibrin fibers?
I don't know the answer to that. My guess is no. The neurotoxin generally comes from like the environment and those kinds of things. Like you're exposed to radiation, you eat various things. There's probably neurotoxin in my Doritos. I don't really care. I'm eating the Doritos. Sorry. People say life is short, life is long, and I want my Doritos. I need some small measure. I don't think so. The concussions do a whole bunch of other different things, but probably not that. In those kinds of places. It's a good question. Any other ones, guys? Okay. So what I'm going to do, we're going to start what happened? There we go. We're going to start the other one, but then we got about 10 minutes and then we'll take the quiz. And I apologize. I did not realize until this morning that I hadn't like actually released this to you guys. So if you're like, where's Dr. Black getting the slides? Why is it not up? There we go. Nope. Maybe it's thinking about it. I turned them on this morning. Okay. As well as the kind of lecture outline note things for this. So none of this stuff will be on the quiz. It's only the stuff that we just got finished. Okay. So we spent a lot of time talking about muscles so far, which is great. Muscles are really, really cool. But hopefully, as I illustrated with the stuff at the end about the fiber type, there's this interaction between the brain and the nervous system and skeletal muscle. And it's really, really important. One, for helping us to determine health and fiber type, but also for how we're able to tell muscles to turn on and generate force. Okay, and so I know you guys have seen all of this before. brain, we're gonna focus mostly and just kind of very obliquely on the motor cortex or the primary motor cortex. We're not gonna talk about the premotor cortex and all of the other kind of neuroscience stuff that goes into this, but basically brain, down the spinal cord, okay? And then in the spinal cord, there are cells that are called neurons. I mean, your brain is, all nervous system cells are neurons, but, well, that's not 100%, but they're mostly neurons. Okay, and there are these cells in the spinal cord that are going to be directly connected to individual muscle fibers in a given muscle. And those are called motor neurons. Okay, they're going to exist in very particular places along the spinal cord. All of them that go to a certain muscle are going to be in roughly the same place. Those cells have an axon, okay? They have like a rope, basically, or a wire that connects them out to those individual fibers. So there's this connection, this hardwired connection to all of those things, and we're going to talk about that. Okay, so brain, spinal cord, motor neurons in the spinal cord, axon out, connect to an individual fiber within a muscle, and that is going to provide this communication. You think contract, right? Brain generates that electrical signal all the way down, and then the muscle executes whatever it is that we wanted it to do. It turns on when we want, it turns off when we want, it gets us the amount of force that we want. It's an amazingly complicated system, but it works almost every time. Okay? On fast, off fast, it's really, really cool and really great. Okay, so if we sort of move on, I covered most of this. Okay, motor neurons in the spinal cord, we oftentimes call them alpha motor neurons. There are beta motor neurons or there are gamma motor neurons that are going to do some slightly different things, but the ones that primarily tell motor cortex to skeletal muscle to contract are called alpha motor neurons. Okay, they look kind of like this. Okay, we'll show some pictures of those in a minute. You have about 250 million muscle fibers in your body. There are only about 450,000 motor neurons. And so you can imagine then, right, that because of this, every motor neuron is going to connect to many muscle fibers. Sometimes 10, sometimes 20, sometimes hundreds, sometimes thousands. Okay. So there's way, way fewer motor neurons than there are muscle fibers. This will become important on Monday of next week. Okay? Every fiber in your body, every muscle fiber has a connection to a motor neuron, and only one motor neuron. Okay? So the cell is the neuron cell is very specific to a given muscle, and every fiber only connects to one. This is absolutely important because if it didn't, movement wouldn't work. Okay? Movement wouldn't work. So the number of fibers connected to a motor neuron is going to determine the size of what we call a motor unit. The neuron and every fiber that it connects to in the muscle, that is collectively called a motor unit. Okay? It's a unit because when that neuron says contract, all of the fibers that it connects to are going to contract. So they work together. They work simultaneously. Okay? And this pattern, one motor unit gets activated, then another, then another, then another, is arguably the most important way that we control and grade out how much force we get. Okay? You just intuitively know how many motor neurons I need to activate the correct number of muscle fibers in the given muscle to get whatever force you want to get. You guys ever pick up a box that you thought was full and you thought was going to be heavy and there's nothing in it? You get too much force and you go that way? We estimate every time we see something, we estimate how many motor neurons to turn on. So how many fibers to turn on to give a certain amount of force. And we're really good at doing it. But sometimes we get it wrong. Okay? So that's what we're going to get into a lot of on Monday is how that particular process works. Okay, last thing, and then we'll let you guys take the quiz, okay? Here is a motor neuron and its axon, okay? Neuron is here. This piece up here is called the cell body or the soma, okay? So the primary cell body of the motor neurons are located in your spinal cord. They're in your spinal cord, okay? Connected to the cell body are these little projections or little fin-like structures around them. Those are called dendrites, okay? Dendrites are the feelers, in essence, of the cell body. They exist to gather signals and information from the surrounding neurons, and then use that information to tell the cell body what to do, okay? So we have to pick up signals coming from the brain through the dendrites to tell the cell body, okay, we need to turn on, okay? Then there will be one long structure, large sort of connection, one large kind of wire, if you will, that comes off of the cell body and will go from your spinal cord all the way out to wherever that muscle is, and that's called the axon, okay? The motor neurons that go to the muscles in my toes are located right about here, okay, in my spinal cord. So there's an axon, or many axons that run from here out of the spinal cord, down, and they run all the way down to my feet. So they run several feet down, okay? What you guys see or saw in anatomy class, when you're like, oh, look, right, my ulnar nerve is right here, or my tibial nerve is back here. What we think of as nerves that you can see in various places are actually bundles of axons, okay? The structure of like a nerve is really just thousands of little axons all kind of sitting together in the same place, okay? It still kills me that we can fall over and not like rip all of our nerves to shreds. You see them right when you dissect them, like they're just little bitty tiny little thing. And you think of all the times you've fallen over or gotten bumped into or whatever else it is on those things, and they tend to be fine for the most part, right? So that's what's going on. On the axon, there's a couple of things that I want you to know. I'm sure you guys have seen all of this before. There's this fatty tissue that surrounds the axons called myelin. Okay, and it will run from the cell body all the way out to the end. Myelin increases the surface area. Myelin is fat. Therefore, it is not a good conductor of electricity, which I know is going to seem weird, but by having the myelin and then having these little breaks in the myelin, here, which are called nodes of Ranvier, and by having all the sodium potassium channels at the nodes of Ranvier, we actually speed up our ability to transmit electricity down the axon. And that's going to become really important. Okay. All right, we will stop there. You guys can take the quiz, right? It should be open. Please confirm for me that the quiz is open and it works. Yep, we're good. Okay. Do the quiz. When the quiz is over, you guys can go. All right. Monday, we'll pick up with this. We will not have a quiz on Monday over this. But I promise you there will be an in-class assignment. We'll do an exercise in class that we will turn. Okay. Cool. All right.